Thank you. This is a little bit of a special presentation for me. Unusual, I guess, for advisory board members to present. But Mana is based in Israel, and as you know what's going on there right now, they were unfortunately not able to travel here. And so I was asked on Sunday if I could jump in. I've only seen the slides like 2 times or so, but let's see. So what Mana is doing as a company is focusing on machine learning, AI, to discover novel LNPs. They were incorporated in 2021, so not only 2 years ago. And they raised, you might have seen today, there was the announcement for coming out of stealth mode, 19.5 million from various investors. You see listed here, very prominent names. They're right now 20 employees. And we will go into some more details here about the science going on at Mana. The team, some of the scientific co-founders, very well known in the space. Avi Schröder, for example, was in Bob Langer's group, so really an LNP expert. Kira Radinsky, one of the robotics experts, also located in Tel Aviv. And then Yogev and Roy, both the co-founders of the company, very talented people, I will say. But also, what I find astounding is very open to getting advice, right? And you see here, including me, but a very nice advisory board, right, that they brought together, bringing in all the expertise that is required to also translate then these LNPs, right? I mean, we've seen a lot of technologies companies, but in the end, I mean, making a medicine out of this, a treatment, that's when you have to translate this into the clinic, and so building on this already. When you look, and I'm in the Bay Area, okay, in Silicon Valley. When you look at, there's definitely some analogies, right? I mean, it's not an easy task. People say launching a rocket is very difficult, right? It's a lot of engineering. That's actually the very same is true for LNPs. So when you look, the relationships here, right? Building the space shuttle, right? I mean, that's the stability of the LNPs, right? Creating the composition of matter. Launching successful, right? The administration route. I mean, there's definitely different variables here. Fly in the right direction, right? I mean, we all talk about the right target cells, right? And I hear very often target organs only. No, it's the target cells, actually, that we have to look at, right? It's very specific where you want to be. Passing the atmosphere, right? So getting out of orbit. That's the cell membrane transfection, right? And then the last step, to land the astronaut safely, right, on the moon or wherever, that's the endosomal escape, which we should also not forget, right? And then the translation happens in the cell. So how does it all work? I mean, how does Mana do it differently than other companies? So the design by AI, so that's, I will go into more details, right? I mean, and I know right now AI is a hype, right? But you will see here how it really works. And then the build, which means creating the test compounds and so on, it's all done by robotics, right? Testing is done by robotics. And then all this data that is generated is feeding the system again to learn even more, right? Make the AI system even better. So how do they get to data? And I think this is something which fascinated me when I joined Mana as an advisory board member. So they have this ML model, and it can really read out of the literature, right? Out of patterns. So you translate the molecular structure. You translate graphs. You translate images, right? I mean, you always see the mouse, or red, right? And you see here the areas that light up where you distribute the LNP to. So that's all read and then built into the system. At the same time, also internal lab data is generated, right? So they've built all the capabilities to run the models, to run in vitro data, but then also in vivo data in mice and so on. So that's all feeding into the system. The prediction of AI, it's getting better and better, and you see here correlations. And it's not only one parameter, right? Not only the efficiency in combination with the payload. They really look at all the physical chemistry parameters as well, cell viability and so on, right? You can have multiple parameters, right? And this is where AI makes it so much better, right? A human, we can maybe read literature or patent a table, right? And we see, or if you change this structure, then it becomes more active. But now seeing this on multiple levels, right? Not only three-dimensional, even more, having zeta values, which is responsible for the distribution in the body. pKa, right? I mean, as you know, there's different pKa's in the body and so on. The cell viability. All this, right? I think that's where AI can only help us, right? What we cannot capture as a human person. So really bringing this all together, right? Including encapsulation, size of the lipids, right? All this information. I mean, if you think about what kind of data set you can generate for each LNP, it's just unbelievable. And then now you can analyze this. So does it work? So what you do first, validate with something that is already known, right? So what they took is a data set here, and you see about 7,000 LNPs, right? And this data set has MC3 in. And those of you that are familiar with the field know MC3 is one of the lipids that's very prominent. It has been used in the approved Alnylam product Onpattro in 2018. And they also looked at the public domain, right, and with the training set. And then they run the model, and they try to see if they can really find MC3 within this, right? And they ranked it, and it was in the highest percentile, right, of the most active compounds. So it did identify MC3 out of all these compounds, right, and as one of the top products, if you will, or lipids. Now, if you would think about how would this translate, you would potentially only test the top 10, 15, right, LNPs and manufacture those, and MC3 would have been part of that. That's pretty good. And in addition, they looked at some other references from Moderna, BioNTech, Pfizer, you see here down, also with a very high accuracy identified. Now, they don't only want to validate, right, existing LNPs, you also want to discover new LNPs. And so that's when they went on, and it's always the same steps. The data acquisition comes first, then you rank the prediction, and then you test in vivo, right? And here you see an example for novel lipids that are active in the liver. And you see on the right side, and really comparable to MC3, right? I mean, so within using this technology, they were able to identify novel lipids, right, that were then patented as well, that are at least as active as MC3. Now, when people say, you know, liver, fine, right? I mean, we've seen a lot there. It's easy, right? Because the liver is our clearing organ. It's actually not very easy. Safety and toxicity is still an issue also for liver, right? So there's still room for improvement there. But in general, I mean, that's true. Liver is maybe from an organ perspective, right? Cell type in the liver, the most easiest one, right? So they took on and went into something more demanding, the lung. And so, again, data acquisition, right? Several formulations from literature. Then they do the AI rank, and then create novel systems, right? And they test them in vivo. And you see here on the right side already that the little rectangle shows the liver, right? And there's nothing in the lung, and there's nothing in the liver. And if you look here at the chart representing the same thing, you really see extremely selective for the lung, right? I mean, nothing in the liver, a little bit in spleen, but still very, very good. Now, this is the first step, right? And then you use AI to generate new lipids in addition that are then improved. And you see here, oops, sorry, an increase, right? 100 times increase using the AI model to generate new lipids compared to the light blue one, which was the one from the previous slide, right? So you see this dramatic increase now, and you see it also lights up much higher, right? I mean, on the right side in this graph where you see the mouse organs, right? So that's successful. Now, when we talk about extrahepatic delivery, it's a new field, right? I mean, so we don't have maybe data, or at least not published data, right, for CNS and so on, right? And so they explored, can we actually do this as well if we don't have data available? And again, for kidney, this is one example here, they were able to identify novel lipids using the same system, but now it's, and I cannot say too much, right, because that's proprietary for sure, but they use data and look into other delivery systems, for example, and so on, right? So you can compile and learn from non-LNPs, and because of all the parameters that I showed you earlier, right, pKa, zeta value, and so on, now you can compose novel lipids that work actually in these target organ cells. And that's a huge success, right? Because that's exactly what we need, and which is always the criticism, if you don't have the data set, we can't work with that. No, you can, right? I mean, and that's really cool. This map is super interesting as well. So what you see here is all the published lipids, right, that they could identify and where you can get access to in an IP map. So structures are clustered by the patents and what they're covering, right? Now you can, when you create a novel lipid, you can match this with this map, and you can see if there's already patterns covering the space or not. And you see the MANA patterns here that I showed you earlier. They are in this little circle with the three red spots, right? They are outside of other IP. And then for sure you can check this again, right? I mean, through search and so on. But it's very predictable. So the IP can also be a parameter in the generation of novel lipids, right? Which is extremely cool. And as you can imagine, some of the companies mentioned here, big names, they are extremely interested in getting access to this, right? Now the kidney, lung, showing you that there's definitely much more out there, right? And the team is addressing this, and there's just some example indications listed here, but you can think about different payloads, right? mRNA, CRISPR also has mRNA for lipids as examples here. They're exploring this as well, and also in partnerships that they're currently exploring. I think there's a huge space, right? And this, if this can be accessed, I mean, this is like really changing the whole field again. And this is the summary slide. So really, I think what hopefully I could show you that MANA has been able in a very short time frame, right? I mean, to already have huge successes scientifically moving these along. And I know that you would say translation from mice to human, right? I mean, that's the difficult part, right? NHPs and so on. This is non-confidential information, right? I mean, if there's more interest, there's more data that validates this further. I can say that much, right? And you see already a lot of achievements. The seed round, I mean, which was extremely high success during these times, right? As you know, it's not so easy to raise money. And the team is very open to discuss potential partnerships, helping others, right, to generate these lipids using their engine, but also in collaborations, right, with payloads and so on to help this move further. And you can either talk to me after the call, after this presentation and call. Yeah, it's a bit Zoom world. You can talk to me after this presentation, and I can connect you with Yogev. His email is yogev@mana.bio, so you can also contact him directly to have more discussions. And thank you, yeah, for having me here today instead of Yogev, unfortunately. And I can try to answer any questions, but I might have to refer. I cannot see anybody here. No, nothing. Yeah, please. Yeah, so they put a lot of work into that, I can say, to analyze which cells within the lung. It was one specific cell type within the lung, yes. And it's the relevant that they were looking for the specific approach that they were going for. Yeah. But you're right, I mean, you have to look into that, right? I mean, it's as I mentioned earlier, it's a big mistake that very often is done, that people just say, oh, we have something that is lung specific, right? That doesn't mean anything. It has to be specific for the cell type, right? And same for CNS and so on, right? I mean, it needs to be very specific. But that's, again, right, where AI can help you, because we just cannot capture this, right? And so.
How are you thinking about translation across species and to humans?
Yeah, so as I mentioned earlier, I think, and I've been working in the space for a very, very long time as well, delivery in general, also AAV and so on. I think in the end, you have to go to NHPs, right, to show that you have the same distribution there, which is not always easy. We also heard earlier today for some of the CRISPR approaches, right, I mean, the CTO from Prime also said this, that then you need monkey-specific guides, right, instead of human and so on. So it's sometimes only a surrogate, right, for sure. But you have to move into higher species for this. And usually for LNPs, the rat is the initial tox model, right? But then NHPs are the ultimate model that you have to test in. And MANA is doing it and prepared to do it, yeah. All right. Thank you once again. And